Transcription. Transcription means formation of RNA on DNA template. Like replication, direction of transcription is also 5'3". Like replication, base pair rule is also applicable in case of transcription. Only one exception is, instead of thymine, it is uracil, because thymine of DNA is replaced by uracil in RNA. Of the two strands of DNA, only one strand of DNA is transcribed at a time. The strand which is transcribed is called sense strand, and at the same time the other strand of DNA which is not transcribed is called antisense strand. For transcription, the most important enzyme is RNA polymerase enzyme. RNA polymerase enzyme consists of two parts, core enzyme and sigma factor. Core enzyme consists of at least 4 polypeptides, 2 alpha, 2 beta, omega may or may not be present. Other component of RNA polymerase enzyme is sigma factor. Now, what are functions of two components of RNA polymerase enzyme? Sigma factor helps in recognition of promoter region. Promoter region, the region where the RNA polymerase has to bind. So one function of sigma factor. Other function is to start synthesis of RNA. After starting of synthesis of RNA, sigma factor is removed from the DNA. Chain elongation. The chain elongation, RNA chain elongation, is brought about by core enzyme. What is transcriptional unit? The part of DNA which is transcribed at a time is called transcriptional unit. It consists of following parts: promoter region, the region where RNA polymerase binds, is start site, then the part of structural gene which is in fact transcribed, and the region of DNA where RNA synthesis stops, means termination of RNA synthesis occurs. Promoter region is characterized by presence of certain sequence on DNA. It is Tata 80 rich region in prokaryotes and Tata region in eukaryotes. Tata 80 rich region is also called as -10 region. Suppose this is start site, it can be considered as 0, this is -1, -2, so at -10 region this sequence is present. Tata 80 sequence is present. It was discovered by Pribno, therefore it is also called as Pribno box. In eukaryotes, at -25 region, Tata sequence is present. It is -25 from start site. It is also called as Hogness box, after the name of discoverer. Suppose this strand of DNA has to transcribe. First, what will happen? RNA polymerase enzyme will bind to the promoter region. After binding of promoter region, the RNA synthesis started by sigma factor. Sigma factor adds first base according to base pair rule. Suppose adenine base is present on DNA, its corresponding base is thymine in DNA, but in RNA it is replaced by uracil. So A lies against U. Again U lies against A. This newly formed strand is RNA, which is complementary to the sense strand of DNA. Now, on coming to the termination region, which is characterized by poly A region, it has many A adenine sequences in this region. It is in fact the termination region, the region where RNA synthesis has to stop. This is stopped by a factor called rho factor, which shows helicase activity. Helicase activity means it acts on this at this place and breaks the hydrogen bond, so that RNA strand gets separated from DNA.